Lastly, we want to take a look of how do transcription factors, again, respond to the environment to alter transcription. So we already saw this with the lac repressor in E. coli responding to whether there was lactose around or not. But this can also happen inside the body and eukaryotic cells. So it's not only responding to things we eat, but different hormones that are made in one part of the body that need to trigger a gene expression in another part, in other cells in the body. And so there are things called steroids that activate transcriptional responses in different cells. And what is the thing that recognizes the hormone to alter transcription? And so the real question was it a protein that binds to the receptor and then starts, or does it somehow directly recognized by the DNA or the RNA? And so to first answer this question, a group of researchers basically took a look and were able to see how much progesterone was around. So they got this 100% of steroid bound, and that's when no enzymes around. Then they wanted to ask, okay, was it protein? So they added an enzyme called pronase. Remember, ACE always means that it's an enzyme, and it digests proteins. And here now there was a much less bound receptor. They did the same thing for DNA and RNases, but those did not alter the amount of bound steroid at all. So this suggested that a protein is what binds steroid receptors, and that likely causes the transcriptional responses. And so then they wanted to ask, where is this steroid receptor located in the cell? Is it already in the nucleus and then somehow the steroid responds? Or is it in the cytoplasm and then together they transverse into the nucleus? So does it go directly? And so the question was, kind of subsequently, was if you inject a steroid, progesterone, does it go directly into the nucleus? Or does it bind a receptor first, then go into the nucleus? And so this second model here would hypothesize that you need to, it has to hang out in the cytoplasm for a little bit, the progesterone, to find the receptor and then translocate. So they did this experiment. And so they injected progesterone and looked for, in the purple, you can see the cytoplasmic progesterone versus the nuclear progesterone, and then measured where the progesterone was after at different 5-minute intervals. And so you can see that the progesterone did not go directly into the nucleus. It first spent a little bit of time in the cytoplasm, then we got a lot of it into the nucleus. So this then suggested the model that there are those protein steroid receptors that are in the cytoplasm. They bind to the steroid like progesterone, and then that gets them to go into the nucleus, where then they can find their activator regions, just like we saw in that yeast gene, to alter transcription and then have you respond to different steroid levels.